Good morning or afternoon. This is Mr. Tierney, and we are going to finish the rest of the notes, and so that means different things for different classes. Hour 7, we'll be starting from Objective 3 and kind of going through some of that bacteria stuff, uses of bacteria, gram staining, gram positive, gram negative, and then for my other classes, we'll just do a little bit on things other than viruses and other than bacteria at the very end, which we call viroids and prions. So before we start, though, I thought I'd give you a couple jokes because I'm gone today, and I figured, hey, why not make you laugh at least a little bit? So a virus walks into a bar, and the bartender says, I'm sorry, but we don't serve your kind here. And the virus says, well, you know what? You're not a very good host. And then two bacteria walk into a bar, and they ask to be served, and the bartender says, sorry, I don't serve bacteria here. And the bacteria says, why? We're staff. So I hope that you enjoy those. We are going to pick up here with Objective 3, and this is about identifying which antibiotics to use on different bacterial infections. So what you're going to see here is anytime where I have something underlined, I feel like I've tried to kind of fast track this for you, but those underlined words that you see in red, those are the blanks that you're going to fill in, starting with Objective 3 there. So Objective 3 says differentiate between gram-positive and gram-negative bacteria. I know it's kind of hard to read there, that first one. Sometimes this software gets the PowerPoint a little bit muddled. So that first bullet there says, this feature is one of the key factors to identifying bacteria. It is based on the properties of the bacteria's cell wall and will stain it a specific color. And so it's named after Hans Christian Gram, who was the one that created this method of testing. Now, I don't know if you remember, but on the Davis family report, the microbiology report that you did with the green beans, this was definitely on there. They tested to see, was the bacteria gram-positive or gram-negative, because that definitely determines what kind of medicine, what kind of antibiotics are we going to give the patient. So a couple things for gram-positive bacteria. One, you can see in this picture here on the left that these are cells, and then right here in the middle, this is bacillus bacteria that has stained purple. And if it stains purple, that means that it is gram-positive. I just kind of remember purple positive. And this means they have a very thick cell wall layer of peptidoglycan, can be treated with penicillin-type antibiotics. That means, of course, penicillin, but it also means like amoxicillin and ampicillin. And so here's your example here, Streptococcus pyogenes. And if you remember from our previous notes, strepto means in chains, and coccus just means a round shape. Okay, so this causes strep throat. And so, of course, I have a pretty gruesome picture here of some really bad strep throat buildup that when you say and they use that tongue depressor at the hospital or at the doctor's office, this is what they're searching for. Now, this is a really bad case, but hopefully you kind of get the gist. So this is gram-positive bacteria. Gram-negative, on the other hand, they stain pink. And so what you see here, sometimes we say pink, sometimes we say red, and this has a thinner wall of that peptidoglycan. And okay, so why is the cell wall important? It's important because that's what the antibiotic is going to break down. Different antibiotics will break down thick walls, and other antibiotics will break down thin walls. So these thinner walls, they actually have another envelope around them. And so you cannot, third bullet there, you cannot be treated with penicillin-type antibiotics, okay? So this might mean like tetracycline, is a well-known one that is not in the penicillin group. And E. coli, the one we just did with our zone of inhibition lab, this is a gram-negative bacteria, so you'd have to treat this with other kinds of antibiotics. So that's gram-negative and gram-positive. Purple positive, pink negative. So what I'm showing you here is just the difference in the cell wall layers. What we're really after is the pink layer, that peptidoglycan layer that's right here in the center. And you can see it's got two labels right here. On the right, we have this gram-positive layer that is extremely thick, okay? Over here, we have a really thin layer, but you'll also notice we have this completely other outer membrane here that certain certain antibiotics are not able to break through. Again, you need to test this because I need to know if my antibiotic is going to break through this kind of wall or if it's going to break through this kind of wall. Again, purple positive, pink negative. Purple positive, pink negative. So let's move on to objective 5 here. You can again see where my underlining is. Hopefully aerobe or aerobics is a word that you've heard before. Aerobic just means that we need oxygen. We are aerobic organisms, and so we breathe in oxygen, we breathe out CO2. So an aerobe needs oxygen. We call this, of course, respiration. The opposite of that would be anaerobe. You see this prefix here, a meaning or an meaning not. So anaerobic means does not or doesn't need oxygen, and we call this fermentation. I'm guessing this is a word you've heard before. We use fermentation in making bread, we use it in making sauerkraut, we use it in making alcoholic beverages. Fermentation is the process where oxygen is not required, and this is also what that Clostridium botulinum did from the Davis family in those canned green beans. It fermented, and so that's the term there we use with anaerobic. Respiration goes with aerobic, and fermentation goes with anaerobic. And then you've got this word right here that if you want to like put a box around it, or I have another blank down here that with the same word, but that word facultative means that they can choose. And so you see that I have this word down here. I put that, or they function in different ways. What that means is they can switch, which is pretty darn cool if you think about it. They can either have oxygen or use oxygen and respire, or they don't need oxygen and they can ferment. They can do both. And so these survive extremely well in the environment. Okay, moving on. So here's some examples. Aerobic would be Mycobacterium tuberculosis. Again, this causes tuberculosis. And anaerobic, that hopefully should be familiar to you by now, Clostridium botulinum, which causes botulism. And then depending on the strain of E. coli are an example of facultative. They can either be aerobic or they can be anaerobic. Just kind of depends on the strain and what the environmental condition is like. Let's explain the different types of bacteria. Now, I think there was a little typo in here. That last one should have been 4, number 4, and I think it was labeled 5 in the notes. But hopefully we're all on the same page. You should be on objective 5 right now. Explain the different types of bacteria reproduction. Asexual reproduction. Asexual reproduction is actually, in bacteria, is a lot simpler process. It's just called binary fission. And so binary fission doesn't have all those stages that mitosis does, like prophase, metaphase, anaphase, telophase. Basically, this is a simplified version where it basically has one copy of the chromosome, it makes another copy, so you see kind of 2 jumbled chromosomes right here, and once it duplicates, it splits. And guess what? After that, it duplicates its DNA, and then it splits. Duplicates and splits, duplicates and splits, over and over and over again. So one bacterium splits into 2 cells, kind of like mitosis. They are clones. So again, this is the parent cell. These are called daughter cells, and each daughter cell is a clone of the parent. Again, this is an example of exponential growth. That the bacteria on your hand, on your clothes, on your desk, if it continues to grow there, it will grow exponentially. It will double every generation. Monerian reproduction, as far as the sexual version goes, we call this conjugation, this word conjugation. That means sexual reproduction in the microbial world. And we use those things, if you remember from the beginning of the notes, called pili. They're those hair-like projections. And so you can see this top picture right here, this top panel. There is this kind of reaching pili right here that is making this bridge between these 2 bacteria. And they're actually exchanging DNA through this bridge or across this bridge. And it's a survival mechanism. It allows them to change their DNA. So if you take a look at this kind of big picture right here, you'll see that part of this plasmid is first off unwound, and then this pili is kind of separated, are meant to be right here in the middle where they're connected. And you'll see that they exchange each other's DNA. This bacteria will have blue and then purple on the middle, and the other one will have purple on the outside and blue in the middle. So they'll kind of mix and match their DNA to hopefully better survive, have some new genes, have some new traits that will make them more fit for their environment. And then here's the weirdo, okay? Monerian reproduction, another kind of sexual reproduction is what we call transformation. And this is when bacteria will take DNA from another bacterial cell that's already died. It's just kind of a waste. It's just kind of sitting there. It's already dead, but there's still DNA molecules in there, and so it wants to use those, incorporate it into its own chromosome, and it will also change its genetics. So this is our dead cell here. You can kind of see this has all been cut up. For the $1,000 question, if you remember what cuts this up, what cuts DNA? What are those DNA scissors called? I've asked you that maybe last week. But bacteria make these. I'll give you a second if you want to check your neighbor. What are these things that cut DNA? If you said restriction enzymes, give yourself a pat on the back. Nice job. So you can see here that this little piece of DNA is then going to be incorporated with a living bacterial cell, and then it makes it a part of its own chromosome, changing its genetic makeup, making it hopefully more adaptive, better able to survive in its environment. So some roles, just real quick, explain the different uses and roles of bacteria. They help us digest our food. You have bacteria inside your stomachs, and they help you digest your food. It's a symbiotic relationship. They get what they need, you get what you need. So help animals digest their food. I'm going to go through these pretty quick. They're used in the nitrogen cycle. Key role in the nitrogen cycle as nitrogen fixers. Plants use these all the time so that they can get the nitrogen they need from the atmosphere. We can't just breathe in N2, which you see right here. That's just floating around in the air, actually more abundant than O2 is. But we can't just breathe in N2. We have to break it down and incorporate it into a useful form. And so you actually need nitrogen in your cells too, and you get those by eating plants, eating salads and broccoli and vegetables and fruits. We use them in environmental cleanup. So what you see here is this oil spill, and we actually have bacteria that will eat oil and decompose the oil and kind of turn it into a safer cleanup. So when the BP had that big oil spill, they actually had planes that flew over and dropped this certain kind of bacteria that helped eat up the oil. So we use them as environmental cleanup. We also use methanogens that can be used to clean up groundwater. We use them in food production, like cheese and yogurt and cottage cheese, sour cream. And a lot of times if you have a yogurt that you eat in the morning, if you look on the back, it will actually tell you what kind of bacteria they use to create the yogurt, what kind of cultures. And we use it as antibiotic resistance. Hopefully this looks a little familiar to you. This is a zone of inhibition test, and you can see these are all different antibiotics, and there's different size circles around here, different zones of inhibition that tell us which ones work the best. So antibiotic production, bacteria and some fungi can actually make their own antibiotics. So I'll give you a little bit of time here. This is for, again, 7th hour. We need to know some differences between vaccines and antibiotics. And this is another slide that kind of came out a little funky. So this first word right here, this is treatment for viruses. Okay? In general, vaccines are treated for viruses. They're taken as a preventative method. They cannot help you once you have the disease. So you got to take it before. Viruses can become resistant to these, but it is usually not a very fast process. It takes a long time. And we produce these. Human beings produce these. On the flip side, antibiotics, they treat bacterial infections. You take it once you already have the disease or once you're already infected. You cannot take it as a preventative measure. In other words, I can't go to the doctor and say, hey, can you give me some amoxicillin antibiotic? I don't want to get sick in 3 weeks. That's not how it works. It's after the fact. Bacteria can easily become resistant to these. They're very, very capable of being resistant to antibiotics, and bacteria and some fungi produce antibiotics. Anytime that if you need to slow down or pause the video, please feel free to do that. I should have said that at the beginning, but if the sub needs to pause so that everybody can write down what's underlined in red, that is perfectly fine. So here's some gruesome pictures that just kind of show you some bacterial infections, how bad they can get. And we don't really think about them being really bad, but of course this is talking about, we're getting to the point where this person might need to have a limb amputated even, that it's that far damaged. Antibiotic resistance. Antibiotic resistance is an increasing problem caused by, you can take them when you aren't sick, doctors just used to prescribe antibiotics all the time, and they didn't know what you actually had, but they just said, well, maybe we'll throw an antibiotic at it and that'll work. Well, the problem with that is if it doesn't work, it makes all of the bacteria in your body more resistant to that antibiotic. Receiving them for things they can't treat, and leftovers ending up in landfills. The other one that I talked about is people who don't take their full medication. If it says, I need you, you need to take your antibiotic for 10 days, and you take it for 6 or 7 because that's when you feel better, then there's 4 doses that didn't get taken, and that means that the bacteria, you might not have gotten all the bacteria, and those are going to be the strongest ones that are the most resistant. So you see this last bullet here. Within just 5 years of publicly using penicillin, there were many bacteria strains that were resistant to penicillin. I'm going to do this real quick, describe how bacteria and viruses make you feel sick. We read the Crummy article. The big things that you need to know is that they destroy cells, or they can release toxins, or they can just block passages. Passages meaning like neurons. They can block blood vessels to the heart, and so can cause major damage that way too. And I hope you like my picture here. Okay, I believe that this is where we have already taken notes, so I'm going to skip now to the very end, and we just have a couple slides. Starting on slide 49, this is for all my classes. This is where we need to just finish up notes. We don't have very long to go. If you notice, objective 12, we talked about the lytic cycle. And if you remember, that was where we used the bacteriophage, that kind of weird looking virus that looks kind of like a robotic spider almost, and it infects bacteria. And so it's going to land on the virus. It's going to inject its DNA into the cell. The cell makes protein copies out of the virus, and then there's an assembly stage where those parts of the virus get assembled. And once those viruses are made, what they do is there'll be so many that the cell will actually rupture, or we call it will lyse the cell, L-Y-S-E. And that's where the lytic cycle gets its name, just as a little bit of review. This is also, if you want to write this down in the margin, the lytic infection, this is what we call the quick cycle. It happens very quickly, and there are rapid side effects from it. Now, the next one we need to talk about is the lysogenic infection. When we watched the video last week, Mr. Anderson talked just a little bit about this, but this is honestly the one that scares me a little bit more, because you don't know you have the virus when your viral infection is lysogenic. So this first bullet there says the virus DNA is made indefinitely by the cell. And we'll talk about what that means here on the next slide when I show you a picture. The virus places its DNA into the host cell, its DNA into the host DNA, so when replication happens, it copies both the virus and the host DNA. And the host, the whole time, has no symptoms of the virus yet. It just carries on like normal, maybe even spreading the virus, not knowing that they have the disease. That piece of viral DNA that is injected and is incorporated into the host DNA, that is what we call a prophage. Okay, so there's a kind of a vocab term for you. The prophage, again, is that viral copy of the DNA, and I'll show you again on the next page. The biggest point I want to make here, though, is that every lysogenic infection, it starts out slow, but there comes a time when your immune system becomes weak, and you're not as able to fight off because maybe you're sick, maybe there's stress, maybe there's a whole myriad of reasons why your immune system might become weakened. And when that happens, there's this trigger that says, okay, we were doing this lysogenic thing, but now let's turn lytic and actually destroy cells and move into a faster way of affecting our host. So this is the last thing I want you to put. That's why I kind of put it in a box and put a star. Every lysogenic infection eventually will become a lytic infection. So lysogenic, this just means like gives, the genic here means like gives birth or at the beginning, okay? And lyso means lyse. So this is like giving birth or at the beginning of the lytic infection. Hopefully that makes sense. Just a real quick picture here. Actually, what I'm going to do, I would much rather use the next picture, but the big thing I want you to notice here is the prophage. you have this DNA that is injected. Here's the host chromosome, and you can see over here this chromosome is going to have the viral DNA, that prophage, incorporated with it, and then it will just continue to divide like nothing's wrong. It'll duplicate the whole chromosome, including that prophage DNA, and make 2 copies. So let's look at the next picture here. So here's the really good picture. From start to finish, this combines the lysogenic and the lytic infection. What you have here is a phage, bacteria, and it lands on the bacterial cell. Excuse me, there's a phage virus that lands on the bacterial cell. And you have this brown chromosome here in the middle. And if you notice here, this little red spot, that's our prophage DNA, and it just gets incorporated like it's just always been there. That DNA has just always been there. And then that chromosome will replicate. And when it replicates, after that, binary fission takes place, and you have these 2 divide. And so now there's 2 copies. And guess what? It happens again. And now there's 4 copies, and then 8, and then 16, and 32. And we have exponential growth of cells that have a prophage in them, or have viral DNA. And then, let's say right here where this arrow is, let's say you have a big stress on your body. Maybe it's that you worked out too hard, or maybe that you were out in the cold for too long. You're susceptible to another infection. Well, what's going to happen then is all these cells that were just normal are then going to say, okay, turn lytic. And so all these viral parts are going to be made, and basically the virus just does this major overhaul takeover of the cell. It'd be kind of like if you were at a Coke factory and I came into the Coke factory, I said, hey, start making Pepsi. Or it might even be weirder than that. Like, let's say you went into a Nike shoe factory and you said, and the virus went in there and they said, you're not going to make Nike shoes anymore. You're going to make Twinkies and Hostess cupcakes. They're weird, and they will do this rapid takeover of the cells and use it to make their own parts in this cell factory. So then you can see they're getting assembled in this row, and they're all being lysed right here in this bottom row. Okay. Last 2 slides that we need to talk about is that there are some things that are even smaller than viruses, and these really kind of scare me because they are not living. They can reproduce, but it's just a molecule. And so this first one we call a viroid, V-I-R-O-I-D. It's just a strand of pure single-stranded RNA. Most of these are plant diseases. They don't have a capsid at all. It's just a strand. It's just a molecule that if it's incorporated into the cells, disease will happen. And so what you see here is this picture of the oblong potatoes. These 2 have the viroid disease, and these are the normal potatoes. Okay? They also are kind of mushy on the inside. They're not very fit to eat. There's no capsid on these, and there's no cure. So here's a picture of the potato from the inside, this viroid infection. And then this is an electron micrograph at the smallest level. Right here, this is just one strand of RNA, and that viroid right there is what causes these plant infections. So very small. We don't really know a whole lot about these yet. Thing we need to talk about are another form of really, really small particles that cause infections, cause damage, and these are called prions, and these are just protein molecules. So they affect animals mostly, and our example here is mad cow. Okay, there's no DNA, RNA, or RNA. It's just a protein. And so what you see here is this is a protein that was made after DNA was transcribed into mRNA, and then mRNA was translated into a protein, transcription, translation. And this is the normal shape. But if it's folded weird, so you see these like kind of arrows that are folded differently than you see over here, then it doesn't serve the right purpose. And most of the time, if there's a wrong fold in a protein, it's just discarded. But on occasion, one will fit in a different spot in the cell, in various spots, and it will cause big issues. And so examples would be Creutzfeldt-Jakob disease, and you can see here, this is just caused by a protein. This is a normal brain. You have gray matter and white matter, and then you can see here all this degeneration that's taken place in the brain. And we're talking about major side effects here, like dementia. We're talking about, can even be some weight loss, forget how to use your motor skills. This is very, very debilitating. And then another one over here, this happens in other different animals. We might even call this cachexia, if you remember that word from your crummy article. but this is called chronic wasting disease, and it's basically your body, its digestive cells, they don't know how to metabolize anything you put into it. So no matter what you eat, it tastes good to the deer, or it would taste good to you, but it would go through your digestive tract, and none of it would get broken down, or none of the nutrients would get used or supplied to your cells. And so it passes right through, and this causes you to waste away because you don't have any nutrients. I think that is it. I know that I might have been a little bit long-winded, but I hope that this was helpful to you. I will be back tomorrow, and your study guide is due tomorrow. Again, our test is Wednesday. If there's time at the end of the class, you're more than welcome to work on your study guide. There's also a packet for you to kind of review, and that can be passed out also. So thank you very much, and I know that this wasn't the best way of listening, but again, thanks, and I'll see you guys tomorrow. All right, bye.